Raquel,
Carolina,
Maria,
Margarida,
Beatriz,
Catarina. We're this group of girls of the 12th grade
from Colégio São Francês,
and we are participating in the Moby Dick project.
The Moby Dick project tries to do an inventory of the biodiversity in rocky shores, in this case from Oporto.
We are doing this tutorial in order to show you our procedure.
We are hoping to see you in June.
Bye!
Our task within the Moby Dick project is to investigate the marine biodiversity of rocky shores throughout time. We need to make inventories of the species present at least 2 times a year, once during summer and once during winter, during periods of low tide, in order to register the presence of all the organisms present. In Oporto, the tide shouldn't be higher than 0.6 meters.
Before Moby Dick field inventories, you must acquire some expertise in the identification of species.
First, select a group of potential species to our shores. You can start with an herbarium collection from your local marine investigation centers. If there's no reference collection yet, that should be the first task.
Second, get some training on the school lab. After collecting some species at the beach, we should start looking at some morphological features. The color can be a good help to divide them at first. Others require microscopic identification.
Not all organisms are microalgae, so you must be comfortable with animal identification as well. There's a lot of taxa to discover in rocky shores.
Now that you are embodied with Moby Dick mission, let's start on the beach. In order to proceed with your field inventories, there's some material you must prepare in advance.
A 50 by 50 centimeter square quadrant divided in 25 smaller 10 by 10 centimeter squares. A device for measuring width and height of our intertidal area in order to get a substract profile.
A field sheet in order to register the observations. Besides registering the common and the scientific name of the species, we must quantify our observations.
Field guide with the most common species in our rocky shores. Moby Dick guide of species was chosen according to some criteria. First, they represent the most common species of local communities. Second, for their ecological versus economical value. Third, for being good environmental indicators. Fourth, for being easily identified at the beach. Once in the beach, you must follow Moby Dick procedure.
Establish some transects perpendicular to the coastline. Identify spatial patterns of abundance and distribution along the intertidal coast stratums. Algae, mussels, and barnacles often dominate different zonation along the intertidal.
In each transect, record height and width of each identified stratum. Randomly place the 50 by 50 centimeters square in the hub, avoiding tide pools or cracks of rocks.
Record the percentage of coverage for sessile species like algae, mussels, or barnacles, and also the density of species for motile organisms as limpets.
We must not forget to apply 3 or 4 replicas in each stratum. The replicas are very important to data validation.
After recording biodiversity, you must record the inclination, the orientation, and the degree of exposure of your sample location. Some organisms adapt better to some areas than others.
Before returning to school, you must not forget to
search for species in some tide pools or cracks of rocks. Use the special habitat field sheet to register the presence or absence of species.
We must also collect some data from anthropic pressure activities. Use this specific field sheet to register the presence or absence of litter, dead animals, sewage, fishermen, or other kinds of activities that can have an impact on the intertidal biodiversity.
In the school lab, enter the data in Moby Dick database. Here, you can follow species diversity and abundance along the years. That is what monitoring is all about, isn't it? At last, don't forget to have fun. For further questions, contact us.